Great to see you back here at the Code of Life lecture. Today we want to talk about identifying changes, or the so-called variant calling phase in the reconstruction of the genome. So again, recap this picture. We have already talked about the DNA sequencing, how the sample is digitalized, then about the alignment, how to get out of the short DNA chunks a longer genome using a reference genome. And now we are in the phase of the variant calling. Variant calling, the purpose of this phase is the identification of genetic variants. Bear in mind, we have talked about this earlier, variants are not necessarily mutations. Mutations have functional impact, variants are somehow changes, however, they are not necessarily have a functional impact. So the input of this phase is, of course, the output of the last phase, the mapped DNA reads, and again, we need the reference genome. And in the end, you will have a long list of variants where somehow alternations are between the individual patient and the reference. And bear in mind, if you have a certain region, you will not have only one read at this region. Due to the mapping and due to the used technology, you will have read this kind of DNA multiple times. You can remember the amplification process. And therefore, reading this multiple times will give you also multiple reads which map to the same or similar position. That means if you have multiple entries at that position, this is called the read depth at this position, gives you the number of nucleotide bases which stores information about it. So how does this variant calling phase look like? First of all, you get the alignment, the aligned reads, multiple of them, the reference genome, put them into these following phases. First, you will do quality filtering. Of course, we have talked about the output of the alignment and the sequencing phase. They will still carry the information how good or bad a certain region or a certain base was read. This is information which is directly taken from the sequencing device. If you have clinical environment, you want to have a certain quality assurance. Therefore, you will filter everything out less than a certain threshold. The next phase is the so-called base pileup, where you will find out which are individual characteristics of a certain region, and then followed by a so-called genotype calling to identify the most probable representation at that region. So this involves a lot of probability measures. And in the end, you will assemble the results. Let's start with the first phase. It's the quality filtering. And therefore, we need to investigate a little bit the results from the alignment. The alignment results will have something like a read, where you have this mapped positions. You see it in this line, like it's still amino acids, A, C, G, Ts, and then you have a so-called cigar string. The so-called cigar string holds information about is there a match or probably a match, or is the algorithm not really able to match it one-to-one. And you see it starts with 2D, and if you do a look up at the cigar string information and the representation, it says D is the information for deletions. You see, therefore, there are some dots here. And then you have a region like 8M. 8M is interpreted as 8 positions where it could be a match or a mismatch. And this is the challenge. It's not really necessarily indicated that this is a region where a mismatch is. You have to now investigate all of the region where you have a match or potential mismatch. Therefore, we filter all other regions off. We filter off the regions which have a certain quality threshold. And then we start to investigate only the regions where you have these M indicated in the cigar string. The second phase is the so-called base pileup. Therefore, again, at the top you see the reference. This is typically the human reference genome. And then you have at the next line the first read of this patient. And you see, again, we have these two missing positions at the beginning because this might be matched later on, or there's a gap here. And then you see a position here which is red marked, the T. The T differs from the reference. So in this particular read, this indicates maybe there's a variant, but it's not necessarily that there's really a variant. It could be also one part of information from the mother's side and the other from the father's side, right? So we need more reads to be aligned at the same region. Therefore, we have here some additional ones. And you see, again, they are aligned differently. It's not necessarily that they match in the alignment. So it could start earlier and end earlier or start later. And if you put them all together, you see some differences. And let's keep at this region where the A is in the reference, and we found this first T. And you see there are some T's red and some A's are red. And you see in the end there is something like this allele representation. It says A and B. A is typically referred to what is in the reference, and B is what we found here. And you see there's one A in our sequence which matches the reference, and most of our representation indicates a T. So we have something like 7 to 1 that there is a T in this region. Typically you would say the A is a read error or something like this, therefore we would decide this is BB. That means this person has really only this single representation T, which is not matching the references. Then you have another region here in the middle where you see 4 times A and 4 times B. Let's investigate it. There you have in the reference, we see the R. The R indicates, it's like a placeholder, it says A or G. So the reference genome may contain other kinds of information like placeholders, like N stands for any or R stands for A or G. And again, you see if you are going through all the reads, we will figure out there are some A's and some G's in there. Typically this is an indicator if you have this kind of equilibrium 50-50 that there is some portion from the mother side or the father side, right? So therefore this is the representation here also in the allele that you have both alleles in this sample. And again, in the first region here, you have again the C in the reference and you have only one T in one of the reads. And you would typically say this one single read is a misread location or the quality is bad at that position. Therefore, you do also the higher read depth, the better the results will be. So what we found out here in this brief example, you have 2 regions which most probably have a SNP. So these SNPs are indicated because we have a higher value of changed base pairs at the location than the reference, right? This is the position 2 and the position 6. So what happens now with this outcome? What do we need to do for that? Now we investigate all of the locations where we found something different. And for that location, we have to do some statistics. And typically there's something called the Bayesian theorem, which is used to calculate the probability for a certain position. The statistical model is depicted here on the right-hand side, and it's not so important for you to get this fully understood in very detail. What is done, we want to computate the genotype probability for a certain region based on the historic decisions. So typically what you will do, you will calculate the likelihood that there is a certain representation, for instance, a T at this certain region. And this takes into account all other genotype probabilities which have been found earlier. This is used typically to smoothen a little bit the results. Again, we are talking about a process in the wet lab where a lot of mistakes or noise can be introduced due to the process. Then we have devices, DNA sequencer, which are not 100% accurate, right? And then we want to do a matching, which is still a probable matching. And now we need this kind of probability check to give you some reduction of noise and poor reading quality. In the end, this helps to improve the mapping and the variant calling phase, and therefore this is typically used in nowadays setup. The next and the final step is the results of the assembly. Now we found for certain regions variants, we decided the probability for them and decided that this is right at this region. What is done now, we assemble all this information in a file called variant calling format. These VCF files are very structured, they have a very limited amount of attributes, you see them in the down here. However, it's a so-called extensible format. That means you can add any kind of attribute value pairs which might be relevant for you by adding this information to the header and put them here in the last 2 fields. Let's talk about a little bit of one of the lines. How does it look like? You have a header which gives you some description about the whole content and also a mapping of the formats. Here, for instances, we have one example. You see there's a ID called GT. And the description for that is genotype, and we will expect something like a string here, and it's located at the first position. Down in the output, and this is typically the same format throughout the whole VCF file. You see first the chromosome position. So now we have a variant found on chromosome 7. Then you have a position which is indicating the locus. Then you have an ID. This is already kind of annotation information. This ID could refer to public available annotation databases. You will find the reference. So in the reference there was a T. In the alternative, so what was observed in our sample, there's an A. And then you have a quality and some filter information, and then you see again this info, format, and sample. And exactly this info and format gives you the information what additional attribute values should be added here. To sum it up, In the end of this phase, you will have a huge VCF file which depicts all the variants. And again, all of these variants are present in the sample, but not necessarily have an impact on any kind of function. And it's not clear if all of these samples are really a driver for any kind of disease. So still we are talking about thousands or millions of variants which have to be investigated in the next phase. So that brings me to the next presentation in the course. So next time we want to assess how variants are important for the treatment. Which impact do they have? How to use the worldwide scientific knowledge publications to find out if this variant is associated to a disease and how this can be involved in the course of a treatment. Therefore, stay tuned, be part of our OpenHPI learning community, and I'm looking forward to have you here next time.